Hello friends, I am here to discuss the physiology questions of GT 182. So let's start with the questions. The question number 42. Question number 42 is, in surgical stress all hormones is increased except adrenaline, ACTH, epinephrine, cortisol and insulin. So let's see the facts about it. So any kind of injury or any kind of stress, write down any kind of stress or injury stimulate multiple centers, like brainstem, hypothalamus, thalamus and cortex. So any kind of stress or injury stimulate these multiple centers. What happened next? The hormones, one of the major hormones, they are catecholamine and SIADH. They are, that is corticotropin releasing hormone. They are stimulated or they are released in the stress from hypothalamus. They are released from hypothalamus, and what they do? They stimulate the ACTH. And the glucocorticoids. And in turns they help to fight the stress. So what happened next? Whenever the stress is there, you need the energy, more energy, that is more glucose to fight the stress. So what happened? The glucagon is also increased or released from pancreas, and in turns increase the glucose level in the blood. So ultimately you get the more energy whenever the stress is there to fight or deal with the stress. Then what happened next about the insulin? What insulin do? Insulin causes hypoglycemia. As we know, insulin causes hypoglycemia. But during the stress, as we just discussed, we need more energy, more glucose, so the insulin cannot release at this point or the stress. So answer over here is insulin. All of the hormones are secreted while the stress is there. That is adrenaline, ACTH, epinephrine, cortisol. The catecholamines itself are known as the stress hormones. So moving to question number 43. Myocardial oxygen demand depend upon. Okay, so let's discuss some points about the myocardium and the oxygen demand of myocardial fibers. So the work done, let's write down the formula of work done. Work done is pressure into volume. So the oxygen demand of a heart or the muscle fibers of the heart, that depends how much work is done by the myocardium. Suppose if the work done is increased, the oxygen demand will also increase. If the work done of a heart is increased, the oxygen demand in turns will increase too. So how does the work done increases? By increasing the preload or afterload. Which increases the work done and in turns increases the oxygen demand of the heart. Apart from the preload and afterload, there is a tension generated. The tension generated by myocardial fibers. And heart rate plus the muscle mass. How much muscle mass is there in the heart, that also one of the factor on which the oxygen demand of a heart is dependent. So, muscle mass. So, all these factors will cause either increase or decrease in the oxygen demand. So, what happened? If the muscle mass of the person is high, the oxygen demand will high too. So, and what happens in anemia? There is a factor in anemia. The last year AIMS exam asked the question about the anemia and oxygen demand. Let's see what happened in anemia. In anemia, there is a change in viscosity or blood viscosity, change in blood viscosity. In terms, what happened? That will decrease the afterload or decrease the resistance. So, the change in resistance, and that in turns will change in the afterload, So, that is how the oxygen demand will change too. Why? Because it is affecting the work done, So, all these factors will cause the oxygen demand change. So, answer over here is yes, preload, which causes the preload, afterload, just we saw, because they both causes the changes the work done. Intramyocardial tension, yes, the tension generated in myocardial fibers, and intramyocardial tension, myocardial muscle mass, yes. If the muscle mass is more, then the oxygen demand will be more too. So, myocardial muscle mass and blood hemoglobin concentration, that is on which the viscosity of blood is dependent. So, in anemia, the oxygen demand is decreased. So, answer over here is all A, B, C, D and E. Moving to the question number 44. 44 says about osteon, which of the following statement are correct? So, let us see some facts of osteon. So, I have a diagram for it. So, this is the diagram of a bone. So, as we can see, there is an osteon. What is osteon? Osteon is the cell on which the mature bone is formed. So, the mature bone is formed made up of, you can write down osteon. The mature bone is made up of osteon. So, over here they show the osteon. The bone contains approximately 4 to 20 lamellae, 4 to 20 concentric lamellae, as we can see over here. The bone contains 4 to 20 concentric lamellae surrounded by the central canal. Write down, surrounded by the central canal, or which is also known as Haversian canal. So, over here the central canal is nothing but the Haversian canal. This is your Haversian canal. So, then what is the Volkmann's canal? There is one more canal. This is your Volkmann's canal. This is also known as perforating canal. So, write down Volkmann's canal or perforating canal. What it do? It connect, write down, it connect the periosteum to Haversian canal. Why it connect? Because the blood vessels are running from the periosteum to the Haversian canal, blood vessels. So all the nutrients whichever are in the blood and which is required for the bone, they are transmitted through the Volkmann's canal towards the central canal. And in Haversian canal there are blood vessels, as you can see. It contains blood vessels, nerves, as well as connective tissue. But what is the difference then? In Volkmann canal, as you can see, the Haversian canal is surrounded by the concentric laminae, but the Volkmann canal is not surrounded by the concentric lamellae. That's the difference in Volkmann's canal and the Haversian canal. So, coming back to the question, they ask about present in compact bone. Yes, they are present in compact bone. So, answer A is correct. The concentric lamellae is present, as we just saw that the concentric lamellae around the central canal is there, and Haversian canal is present. Yes, the Haversian canal is there. Volkmann canal surrounded by concentric lamellae. No, the Haversian canal is surrounded by the concentric lamellae. They are 4 to 20 in number. So, answer of question number 44 is A, B and C. Question number 45. Which of the following sugar are found in amniotic fluid? So, let's discuss some fact about the amniotic fluid content. Amniotic fluid content. Write down. Amniotic fluid content. Amniotic fluid content or composition. What is the composition of amniotic fluid? It is, write down, 99% water and rest are solute. 99% water and rest are solute. What are solutes? Like proteins, lipids, glucose, protein, lipids, glucose, urea, uric acid, creatinine, and some ions like sodium, chloride, etc. So, these are the solute which are present in the amniotic fluid, but 99% is the water content. So, 99% is water content. So, as we can see, only glucose is the sugar which is present in amniotic fluid. Amniotic fluid does not contain sugar except glucose. So, none other than the glucose, all other sugars will be absent. So, answer over here will be option C. Option C is the answer of the question number 45. Glucose is the answer. Moving to the question number 46, saccadic eye movement is controlled by. Let's see first of all what is saccadic eye movement. So, what is saccade and saccadic eye movement? Write down saccade. Question number 46. Saccade. So, write down extraocular eye muscles. Extraocular eye muscles are never stationary. They are never stationary. They are having jerky movements all the time. They are having jerky movements all the time, which is known as saccade. Okay. So, why these saccades are there? They are helping. Write down. They are helping visual perception. They are helping proper visual perception. They are helping proper visual perception. So for that the saccades are present in our eye. So what they do, they are mainly programmed, they are mainly, write down, they are mainly programmed and controlled by frontal lobe, and with the help of frontal eye field area number 8, frontal eye field area number 8 in the cerebellum, frontal eye area in the cerebellum. Cerebellum basically help in gaze maintenance, which is known as in frontal lobe there is saccades are present which help in visual perception. In cerebellum the cerebellum help in gaze maintenance and which is known as vestibulo-ocular reflex. This was the question asked in NEET exam and the PGI in 2011. What is vestibulo-ocular reflex? So let's see quickly what is vestibulo-ocular reflex. When a person moves his head to one side, either right or left, one side, the vestibular apparatus sense this movement or detect this movement, the motion of head via stereocilia in the horizontal, because your head is moving towards one side. So horizontal, that is detected by the stereocilia in horizontal, or which is also known as lateral semicircular canal. What happened next? This motion is sensed by the vestibular apparatus and the info will then go to the vestibular nucleus. This info will go to the vestibular nucleus, which is located in cerebellum, What cerebellum do to maintain the gaze then? The cerebellum causes eye movement. It causes eye movement to opposite side, which is known as vestibulo-ocular reflex. How vestibulo-ocular reflex? It is sensed by vestibular apparatus sent to the vestibular nucleus in the cerebellum. So what cerebellum did the action? Cerebellum caused the eye movement towards the opposite side. If the person moves his head to the left, the eye movement will be on the right side. That is known as vestibulo-ocular reflex. So question number 46, the answer will be frontal cortex over here. The answer will be frontal cortex. Moving to the question number 47. Which of the following are true about SIADH? Okay, fine. Let's see about some facts about the SIADH. As we know, ADH, antidiuretic hormone, it is released by supraoptic nucleus in hypothalamus, which act by two receptor V1 and V2, in response to, when it is secreted in response to, a decreased blood volume, that is hypovolemia, decreased blood volume, or increased plasma osmolality. Increase plasma osmolality. Okay? Then move to the next. Increase plasma osmolality. It act by two receptors, that is V1 and V2. V1 and V2. What V1 causes? It causes vasoconstriction, but V2 by increases the cyclic AMP, in turns, which causes the increased water reabsorption by increasing aquaporin 2, and it increases the water reabsorption, which in turns, what happened to the urine? Urine will be hypertonic urine or concentrated hypertonic urine, because more and more water is absorbed in the body. So urine will be hypertonic. And what happened to the plasma? The plasma will be hypotonic. So as we can see over here, it acts when the plasma osmolality is increased, that is the hypertonicity, and it causes hypotonic plasma. So how much it can cause the concentration? The concentration may go up as high as the medullary concentration or osmolality in the urine, that is 1200 milliosmol per liter. Milliosmol per liter. So let's see which all following are true about SIADH. Desmopressin is used for the treatment. Okay, so what happened? What we discussed about the ADH, what happened in the deficiency of ADH? Deficiency of ADH causes diabetes insipidus. The water cannot be reabsorbed, and the 10% of the water from the collecting duct is removed via urine, because the P cell cannot absorb the water in absence of ADH. So the diabetes insipidus will occur. What happened in excess of ADH? The SIADH will occur. So in SIADH, the excess of water is reabsorbed, the urine will be more and more concentrated, and the plasma will be hypotonic. And desmopressin is used in diabetes insipidus. So it is not used in SIADH, desmopressin is used in diabetes insipidus. The A cannot be the answer. Option B, vasopressin level is inappropriately high. Yes, the vasopressin, ADH is also known as vasopressin, so vasopressin is inappropriately, that is excessively present, so vasopressin level is high. Plasma osmolality is higher than the urine osmolality. No, plasma osmolality is less, because more and more water is reabsorbed. So the plasma osmolality is low, but the urine osmolality is high. Increase osmolality of urine, yes. Increase osmolality of urine. Increase osmolality of plasma, no. There is a hypotonic plasma. So answer of 47 will be B and D. Moving to the next question. Question number 48. Destruction of sensory area 1 of brain leads to loss of which sensation? So let's see some facts about the sensory area number 1. So write down. There are two types of sensation. There are two types of sensation. Two types of sensation. Pure sensation and cortical sensation. Pure sensation and cortical sensation. What is cortical sensation? They occur only at the level of cortex. Occur only at the level of cortex. So They are conscious in nature. They are conscious in nature. Which are those sensations which are felt at the level of cortex? They are mostly which the sensation which are carried through the dorsal column. They are the cortical sensation. Dorsal column sensation. Most of dorsal column sensation, most of, not all, but yes, most of dorsal column sensation, for example, stereognosis, touch, the fine touch, because the crude touch are sensed through the anterior spinothalamic tract. Fine touch, stereognosis, tactile localization, graphesthesia, etc. So, they are the cortical sensation. All occurs at the level of cortex, all sense at the level of cortex. So, where the pure sensation, write down where the pure sensation felt at subcortical level. For example, pain. You can take the pain. Pain can be felt at both the level, cortical and pure. How? Let's see. The pain, just pain, is felt in the thalamus via thalamus. Just pain can be felt at the level of thalamus, but all the properties of pain like but the duration, intensity or severity, the type, intensity, severity, location, all they are felt at. They can be sensed or felt. They can be, more appropriate is sense. They can be sensed at the level of, only at the level of cortex, not at subcortical level. What is a clinical application of this? Whenever we cause the pain in the unconscious patient, he can feel the pain, but he cannot localize or say all these properties of pain. So, write down. So, in unconscious person, unconscious person can sense the pain and react to it or react to pain without becoming conscious. So, let's see the destruction of sensory area 1 of the brain, which is a cortical area. So, answer will be B, C and D. D, that is 2-point discrimination. Pen can be sensed at both the levels. It can be sensed. So, destruction will be if the destruction of the cortical area, that is sensory area number 1, there will be a lead to loss of sensation of fine touch, stereognosis and 2-point discrimination. 2-point discrimination also carried through the dorsal column, but the pain can be felt at subcortical level, so it cannot be lost. Moving to question number 49, the substance freely filter across glomerular capillaries. So, let us see some things about the glomerular capillaries. So, write down glomerular capillaries. They have the fenestration. So what about them? The effective pore size, and they have the pore in that fenestration, that is in the basement membrane. The pore size, effective pore size of filtration membrane is 4 to 8 nanometer, is 4 to 8 nanometer. So suppose the pore is of 4 to 8 nanometer. So whatever the substance are, the molecule, the substance or the molecule which are less than 4 nanometer, they can easily filtered out. So they can easily or freely filtered. They can easily or freely filtered. For example, water molecules. Their size is approximately 0.15 nanometer. The size of water molecule is 0.15 nanometers. Other examples are the ions like sodium, glucose and inulin. Sodium, glucose and inulin. They can be filtered, they are freely filtered. What happened to the albumin? Albumin in the basement membrane of the kidney, there are proteins present which are known as sialoproteins. The protein which is negatively charged protein present in the basement membrane. In the basement membrane, they are known as sialoprotein. And remember, as we know that the albumin are also negatively charged, so both when it come in contact, they repel each other. So due to presence of negative charge, it is repelled and not filtered, hence not filtered. What happened to thyroxine? Thyroxine, write down. Thyroxine, it is bound to albumin and globulin, as we already know. It is bound to albumin and globulin. So this is also not filtered, not filtered. What happened to the molecules who are more than 8 nanometers? They are not filtered because the size of the molecules are more than 8 nanometers. Size of a molecules like, for example, RBC, WBC, platelets, etc. They are not filtered due to size, large size. Okay. What happened to the bicarbonate and chloride? Bicarbonate and chloride, they also have a negative charge, but their size is less than 4 centimeters. So, their size is less than 4 nanometers. So that's why they are freely filtered. Write down the bicarb and the chloride. They also have a negative charge, but the size is very small as compared to the fenestration that has effective pore size. So they are freely filtered. They are freely filtered. So the answer of question number 49, substance which are freely filtered across the glomerular capillaries. Glucose, yes, glucose is freely filtered. Albumin, no, because of the negative charge. Thyroxine, because it is also not filtered. Why? Because it is bound to albumin and globulin. Creatinine, no. The bicarbonate, yes. So answer over here, substances which is freely filtered across the glomerular capillaries as glucose and bicarbonate. Okay, moving to the next question. Which hormone regulate serum calcium level? Serum calcium level. PTH, calcitonin, adrenaline, 1,25 dihydroxycholecalciferol and procalcitonin. calcitonin. So let's see some facts about the calcium hormone, calcium regulation via hormones. So there are let's make a table and make it very easy. Write down PTH, calcitonin and vitamin D. There are three main hormones which regulate or maintain the calcium level in the blood. They are PTH, calcitonin and vitamin D. So write down over here the calcium and the phosphate. Calcium and phosphate. Let's see what happened to calcium and phosphate when these hormones are released in the blood. The parathyroid hormone increase the calcium level in the blood, but they decrease the phosphate level in the blood. So what happened if the phosphate is decreased? This calcium cannot be used for the calcification of the bone. How it happens? They decrease the bone activity. So they decrease the bone activity and they increase the resorption. They increase the resorption via stimulating the osteoclast. Via stimulating the osteoclast. So via stimulating the osteoclast. What happened to what the calcitonin do? It also increases the it decreases the calcium level, but it also decreases the phosphate level. It also decreases the phosphate level. So what happened? It also increases the resorption, or it inhibits the resorption. Sorry, it inhibits the resorption via inhibiting the osteoclast, via inhibiting the osteoclast. Vitamin D, as we know, if we take more and more vitamin D, more and more calcification will be there. For calcification, the phosphate should be increased in the blood level, and then only the calcium appetite will be formed and the calcification will occur. So both will be increased. So bone calcification or calcification will occur, or bone formation will occur. So what about the parathyroid hormone? The osteoclast have some receptor, that is the receptor that is rank receptor. What is rank? That is receptor activator of nuclear factor kappa B. Nuclear factor kappa B. So osteoclasts express the rank, while the osteoblast express rank ligand. That's the only difference, and this question was asked in PGI 2013. So rank ligand. So and what about the PTH? One information about the PTH, that PTH increases the new osteoclast generation, which is known as osteoclastogenesis. So that's how it increases the resorption. So question number 50. The answer of question number 50. Which hormone regulate the serum calcium level? PTH, calcitonin, adrenaline, and okay. So answer over here is PTH. Yes. And calcitonin. PTH and calcitonin.